Okay, good. So I'm working in the Nanyang Technological University. I'm basically the chemical biologist. So as you can see my presentation, the title, I'm going to share with you some other very recent studies in the fluorescence imaging and the theranostic bio applications. So my presentation is, including my research, is okay, is regarding to the rationale of the different imaging modalities so people use in the hospital. As you can see in this scheme, there is a bar, and above the bar that is commonly used the imaging modalities people use in the hospital, and all we know the X-ray, CT, MRI, PET, ultrasound, and so on and so forth. And below the bar, you can see there is a lot of spectroscopy and microscopy things. This is optical imaging. So optical imaging have also been well developed. Even in the Nobel Prize, quite a few awards have been given to the optical imaging. But very unfortunately, by right, so not so popular use in the hospital. As you can see above of the bar, optical imaging in the hospital for the human being, the real time the application is still quite limited. This is why this may require the people working in the areas, try to develop a unified and also the very good imaging props or contrast agents so medical doctor can use for the imaging for the analysis. Of course, so many other device or deep the theories, so people still needed to put more efforts to do the investigations. So for the optical imaging things, now like the other imaging modalities, very useful, but still a few technical concern people keep in mind. So optical imaging can get a very sensitive detection, can also achieve the spatial temporal resolution, but very unfortunately, so far the optical imaging in the real human being's body, when people do the application, some of the barriers or drawback still we have to encounter. So penetration, that is a big issue, and sometimes a lot of irradiations at the short wavelengths or the visible the spectral windows sometimes can cause the photo damage or phototoxicities. So how we can achieve the optical imaging and apply them into the deep tissues and can get the spatial temporal resolution and also precise the detection, but in the deep tissue manners, so that is still the big concern. So people, we wanted to solve the things in the hospital. This is what in our groups in the past few years, we are doing a lot of chemical biology the design, apply the different fluorescence probes, especially we are focusing on the near infrared, the imaging the system, which we hope can solve the issue or at least can improve the regulation and the bioimaging in the living systems. So in terms of the near infrared, the imaging the probes, and of course, people develop different type of the contrast agent. And in our groups, so we mainly develop different types of the near infrared probe molecule, including the polymer, including the small molecule probes. But in this presentation, I wanted to share with you very unique nanoparticles which based on the lanthanum night doped inorganic nanocrystals, which people determine as upconvergent particles. These particles have a few very unique optical properties. So basically, they can absorb the light to the light irradiations, same as all the fluorescence molecule. But a very different merit of the upconvergent particles that many can attribute it to these special materials which they can absorb the near infrared light irradiations. And the other very unique merit we are very keen in the past few years, that is after the light irradiations of upper conversion particles, you can obtain the multiple emissions. So multiplexing the emissions can really do a lot of very good advantages, can solve a lot of synthetic and biological the efforts. Okay, this is very unique the stuff for us to achieve the near-infrared fluorescence imaging. Of course, these unique nanoparticles can exhibit very good photostability and not much of the toxicities as concerned by most other people. So we are very keen to use this up-conversion particles to do the imaging, to do the bioregulation, the studies. I'm going to show you a few examples to promote these unique nanocrystal structures. Okay, the first application I wanted to share with you is the light triggered functions for bioactivity the regulations. Okay, basically light people use in the biological system to control the biofunctions and disease treatment. So people already use some, already prove some the concept. So in the market, the typical term for these areas that is the optogenetics. Basically, people they use light to control some light responsive the channels, especially in the neural system. So people they can control the biomolecular activity by the light. So they can regulate the neurological disorder and also can get some disease treatment management. So in the society of optogenetics, so far people they use is mainly based on the normal light irradiations, which is the UV light or the short wavelengths visible light irradiations. So basically, this light irradiation can function on the cell membrane, can promote or can inhibit the neuron functions. So people can understand precisely the activity, the functions of neural cells. So people can have a deep understanding, maybe can have some good strategy to help people to develop a useful drug molecule to promote the neural functions. Even though there's so much so useful, but unfortunately, so current optogenetic strategy mainly based on the UV light or visible light. As I mentioned before, the deep penetration that is issue, light toxicities potentially that is also the issue. This is why we wanted to use a near infrared light to try something for optogenetic control of cell activities. And we still focus on the cell membrane because that is the barriers to function as a barriers to separate the cell with outside of the world and many important biomolecules on the cell membrane. And we wanted to use a near infrared light to control the cell functions. Of course, we are using the up conversion particles. But one big thing is how we can precisely localize the particle on the membrane structures. That is the point. That is an important thing we needed to make first. So in this special design, we rely on the carbohydrate metabolism. So we use the artificial mannose carbohydrate fit into the cell after a few more steps of glycolysis or sugar metabolism. So the artificial sugar somehow can trap on the membrane with the functional group. And then for the up conversion of particles, we just simply do the modification with the DBCO, which is the triple bond on the particle surface. Then based on the simple and specific reactions of click chemistry, the up conversion particle can specifically localize on the cell membrane. So at this moment, if we shine it on the near infrared light, one of the emissions, which is 480 nanometers, can shine on the membrane, so we can control the membrane activity in the highly sensitive and special temporal manner. So with this information in hand, we do quite a few different biological studies. At the very beginning, we needed to fuse channel adoption, the light responsive the membrane protein on the cell surface, which we fuse with fluorescence protein. As you can see, all the membrane have a green fluorescence the signal. That means the gene expression, no problem. And next step, we just localize based on the carbohydrate metabolism. To localize the upconversion particles on the surface, you can see the blue color on the top. And we use the membrane staining protein, we merge each other, confirm. So the gene expression of channel rhodopsin, no issue, particle fabrication is quite okay. Then we can do the precise optogenetic control of cell activity. For the membrane function, we do the investigation is mainly based on the cation, the influx, which we focus on the calcium, and we can see the fluorescence change after we control the membrane by the near infrared light. Fluorescence type, you can see the staining. Meanwhile, so after the cation go into the cell membrane, the membrane potential may change. We use a patch clamp technology, can double confirm the near infrared light can control the cell membrane activities. So the activity control by the optogenetics to control the cation go in and out of the cell membrane, that can cause many other subsequent cell function regulations. One of the activities we focus on the optogenetic control of cell membrane, that is apoptosis, the change. So after we shipping, we control the cation influx into the cell membrane, and they can regulate of apoptosis things happen. And we are using the standard the biomolecule to confirm the membrane change of the apoptosis caused by the cation influx controlled by the optogenetics. And that can really get very precise control. And because of the cation regulations, later on the cell activity can be precisely manipulated as well. That is what we can achieve. And not only simply to prove a concept into the simple cell structures, and more importantly, we also push this technology into the living animals, because one of the unique applications for the near infrared light is the tissue penetrations. And this is why we push this near infrared light optogenetic control into the zebrafish. And we do the gene expression to control the light responsive to membrane protein, and have a function with the upconversion particles. After the labeling, followed by the near infrared light irradiations, we really can control the cation influx happening into the fish body. And importantly, we can also monitor the change of apoptosis. Of course, as very initial studies a few years back, we compared the advantages of near infrared light for the tissue penetrations. As you can see in this scheme, tissues can simply block most of the, the conventional use the light for the optogenetics, but for the upconversion near infrared light irradiations, deep penetrations can be easily achieved. So far, we using the optogenetic tools to do the cancers and also the diabetes disease manipulation, and hope in the near future, so we have more chance to share with you the details of optogenetics based on the near infrared light triggered by the upconversion nanoparticles. We are the batch of the researchers using the near infrared light to approach the genetic studies. So far, many people, very good scientists, also using this technology to do the gene expression, to do the disease control. So not only for the simple optogenetic irradiations to control the cell activities in our groups, we also fully utilize upconversion particles. We really wanted to achieve more, and we wanted to develop the multiplexing the biomarkers, and hopefully we can achieve the real-time evaluation of disease dynamics. For the critical disease, so how we achieve the early diagnosis, that really required very precise the monitoring or tracking of a different disease stage. I'm not saying so detect some special biomarkers, yes and no, less or more. I'm more pretty much wanted to see, we wanted to detect the biomarkers or develop a probe to monitor the biological pathway under the very dynamic situation, means all of the things always change. We wanted to monitor the dynamic in the very complex situation. But very unfortunately, so far in the market, such kind of the probe still remain elusive. So this is why we wanted to develop the unique probe molecule, so they can work as the biomarkers and can simultaneously to detect the disease, the factors at the different spectral windows, so we can get the precise and sensitive the readout. So that is our rationale. And the biomarkers we focus in the group that is free radicals. So that is important signaling. The modulators can have functions contributed to the metabolism, digestion, and the drug, and the therapy, and so on and so forth. And in the bodies, we do have a different type of the radicals. Can be oxygen-based, can be the nitrogen-based, the radicals. And for the people working in the areas to detect some certain single the radicals is not new, and it have been well established. But in our groups a few years back, we really wanted to develop a unique proper molecule so they can simultaneously to read out the different radicals at the same time. And also they can be used to reflect the dynamic change happen into the disease at a different stage. So that is the rationale and the design we wanted to achieve. So technically, in order to make this happen, so we're using the upconversion and nanoparticles. And remember, one of the advantages for upconversion is they have a multiplexing emissions. So they have different emissions out. So we just develop a different image in the probe, which they can have responsive reactions to the, for example, ROS the radicals, this hydroxy 5, for example, and they can have a specific signal change at the 650 nanometers, for example, and the other molecule which they have a specific responsive reactions to the nitrogen-based radicals. And after reactions, they have a signal changes at 800. So as you can imagine, we can really anchor these two fluorescent molecule on the surface of upconvergent particle, and each of them have their own responsive wavelengths to the ROS and RNS change. So we can really achieve the detection of two radicals simultaneously at two different wavelengths. And more importantly, as you can see in this figure, and after the ROS, the reactions, the molecular signal will increase. And after the RNS reactions, the signal of the radical changes can make the fluorescence and the emissions drop. So finally, we can create such a very critical platform with two molecule on the surface. Anytime we have a different ROS and RNS reactions, so we always can get a different fluorescence and absorption change. So can give us very good rissometric analysis to precisely mapping the radical changes at a different time point and a different stage. Okay, and in order to make the signal more deeper penetrations in the body, of course, we focus on the near infrared light window, but to get a signal more specific and the tissue the penetration, we focus on the ultrasonic signal. That is new technology. Basically, the near infrared light can go into the tissue and they can produce a heat. If you heat, you want an expansion into the environment or surrounding the environment, so they can release the energy out. One of the energy we can capture, that is ultrasound. Okay, so what then you can understand what we wanted to do, and we give a light into the tissue with our probe molecule, and we can observe, we can capture the signal of ultrasound. All this can be done under the two different wavelengths. Can really give us very precise the imaging for the disease, the trapping and analysis. Okay, so let me share with you of some in vitro and in vivo the data. This is the spectroscopy change. And this is the fluorescence change after radical reactions. And this is the absorption change of a dye molecule, hydroxyl5 and CVS7. And each of dye molecule have their own specific reactions, have a minimum interference from other radicals. And the signal they change in a very opposite manner. And such detection can be used for in vitro response to the radicals, and also can be used for the living cell, the imaging of different radicals. As you can see at the 680 nanometers window, and oxygen radicals, once you have, they can have a signal increase. And for the nitrogen-based radicals at 800 nanometers window, the signal will drop. So anytime, any locations, if you have a different radical, signal will change in the very opposite and a different manner. So you really can achieve the ratiometric analysis to understand what's happened in the body. Okay. And with this promising data in hand, we move forward, of course, to do the tissue the penetration and to prove the concept of imaging happen into the living mouse. And we do the scanning of the signal to trap the up-convergent particle into the liver, because that is a specific organ to do the drug and some important things, the metabolism. So all the radicals may happen over there. So we wanted to scan the liver to understand the radical changes and also to map in the disease change at a different stage. Okay, as a proof of concept, we inject a mouse with some special molecule, for example, lipids, which we already know can produce more ROS the radicals. As you can see, indeed, we can achieve the 680. If we inject the lipids, more oxygen radicals can be generated. We also do the separate animal models with different drug molecule to induce different radicals, including the drug molecule EP can only induce or predominantly induce nitrogen-based radicals, for example, at 800. So we really can use our probe, can see if a body have more oxygen or nitrogen-based radicals, we can precisely to know them, to monitor them. Okay, so not only can do the detection of ROS or RNS in the body, importantly, we can also do some special drug screening and also to understand the drug metabolism. These are two examples. One is the RNH drug, which people use in the hospital to treat the pneumonia. And the THA is the other critical drug molecule people use in the hospital to treat Alzheimer's disease. But very unfortunately, a few years back, recently, Tacrin have been withdrawn from the FDA list. People they see they don't know the reason. Both of these two drug molecules can cause toxicities in the liver. If you use a traditional biomarkers, protein or enzyme, you can only see the toxicities, overexpression of protein or enzyme. You don't know the reason. You cannot tell any difference. But if we are using our proper molecule to do the scanning, different radicals at a different time point, as we can see, if we scanning the two drug molecule, let me show you the figure. This scanning can simultaneously proceed at two different wavelengths, as I mentioned before. And the whole wavelength range, as you can see, the THA mainly can cause oxygen-based radical response. For the RNH, isocyanate drug molecule, they're toxic molecule, but you can see the metabolic profile, so they can mainly cause nitrogen-based radical change. So you may ask, hey, what's happened for the radicals, nitrogen-based radical predominant or oxygen radical predominant? Let's go back to the normal life and the pharmaceutical chemistry or the drug metabolism or food metabolism. All we know is the drug molecule in our body, they will go through the phase 1, phase 2 metabolism. Okay, if drug molecule they go through the phase 1 metabolism, so confirm that is will go to the redox reaction. That's all we know. If drug molecule they only go to the phase 2 the metabolism, so that is mainly based on some conjugation, make drug molecule smaller size or more hydrophilic, easily can be cleared from kidney. So let's see if we find the tacrin, they are pretty much based on the predominant signal changes at the oxygen-based radicals. So we just mapping the P450 enzyme. We know potentially what the reason and which of the pathway of the P450 can deal with this molecule. For the RNH, even though they can cause the toxicities, but the remaining because of some other the unknown issues, because they may not go to the main phase 1 drug metabolism. That's what we observed before. And more importantly, we just make the bar of our scanning, doing some simple calculation, and above the bar that is oxygen radicals, the scanning, below the bar that is nitrogen-based radical, the contributions. And we can see at the disease at a different stage, radicals may change in a very different manner. But if I'm using the traditional biomarkers, you don't know, cytokine or some other AST or GST the enzyme, which people use in the hospital to reflect the toxicity of liver, either there is toxicity or there is no toxicities. You cannot see the difference. But using our Prof molecule, you can dynamically to see the disease at a different stage. So that is our contribution. So we can use the upconvergent particle to scanning the different radicals at the different time point simultaneously. And also we can precisely to understand the drug metabolism, and we can achieve early stage of disease diagnosis. Not only to screening the different inflammation or different stage of disease, and also we can using the same concept to evaluate the therapeutic, the treatment effect also happen into the dynamic manner. So you don't need every time just as a traditional way to draw the blood, to test the blood sample, to do the liver biopsy, painful, take time, tedious. Supposedly, we can simply scan the radicals by using the fluorescence imaging or the photoacoustic. Based on the radical changes, you really can get a lot of very important, useful, but very easy information. So at this moment, so we do the other design. We're still using the upconvergent particle. We still screen the radicals, and we are only screening the ROS, the radicals. And we wanted to evaluate the change of ROS change to see the disease treatment in the liver happen or not. We also can screening of the different drug molecule for the efficient activity happen or not. And at this moment, we use this star molecule before and after the radical changes. They can have a shift, only one star molecule, but before after the ROS reactions, they can have a blue shift from the 800 to the 680. So we still can get the ratiometric analysis to precisely understand the therapeutic effect. Let me quickly show you the example. And this is in vitro study. So radical change is very specific and only can respond to the hydrogen peroxide. And the spectroscopy absorption change because of blue shift. You can get a two spectrum window as a ratiometric analysis. Upconvergent emissions also can change accordingly and can get the reverse ratiometric analysis, different from previous project. And importantly, such imaging reverse ratiometric analysis can be achieved in the living cells based on the photoacoustic and also fluorescence the imaging analysis. And such concept also can be used for the living animals. That's even more important. And the drug molecule we are using, that is RNH, which can cause a toxicity of liver, I mentioned before. Okay, can cause more or less of radical changes in the body. And after we produce this toxic model, we added different antioxidant drug molecule to see how the drug molecule can improve the liver function to minimize toxicity caused by the drug molecule. And you don't need to draw the blood simply based on the scanning of the fluorescence. This is fluorescence modality, and this is a photoacoustic the analysis. And based on ratiometric analysis, you can see. So this is a reverse ratiometric analysis based on the two spectrum window blue shift of a dimolecule after reactions with the radicals. You really can get a precise signal changes. Easily can tell us. So after the treatment by using the drug GSH or NEC, so the radicals can be improved or the toxicity can be improved, the toxicity, the treatment can be improved or not. So we can even do the drug screening, and the radical changes because of the toxicities happen into liver. You can see very high signal, and after we added the antioxidant drug molecule, so signal can drop. So that is the things we can achieve. More importantly, for the radical changes, same as before, radical changes can happen at very early stage, much earlier than the AST or ALT, the enzyme which people they typically use as a standard biomarkers to reflect the liver functions. So the radical changes can be much faster in the liver than the biomarker of the protein changes in the body. That is what we can achieve by using the up conversion as a platform, as the paradigm to evaluate of a therapeutic effect, which happen in the dynamic manner. Okay, so finally, I wanted to give my summary to share with you a few simple story up conversion. The platform, particle platform can be used as a near-infrared, the responsive the paradigms, so we can use for the optogenetic control, can use for the simultaneous screening of different radicals to achieve the evaluation in the very dynamic situation, and also the situation can be also applied for the disease treatment. We can do the very efficient evaluation, no need to proceed a very tedious and expensive the testing process. Okay, so finally, I'm using this chance to appreciate all my students and the staff working in the lab, and also appreciate some of my collaborators. And importantly, I wanted to thank the grant agent give us the opportunity to proceed such not cheap the imaging study and the research. And finally, so by using this time to share with you of a nice slides in Singapore, and hopefully, so the presentation can convince you, and Singapore so also can convince you come over in the near future, so we can have some collaborations and to proceed something important in the societies. Okay, thank you so much. That's all my presentations, and by these opportunities to do some the advertisement for my staff. Yeah, thank you so much.